Log on, tune in, find out. Another good idea from Cambridge.
Okay, so Tim will talk about genome sequencing.
Is the mic working? Yeah. Okay. What I'm discussing here today is kind of a different approach to phylogenetics compared to a lot of the other talks. So rather than applying theory to a specific set of data, we've got a very complex and messy set of data, and this is in a sense a biologist's take on grabbing bits of theory that might give us some sort of insights into the data. So here I'm discussing these Leishmania parasites that infect people across the world, and what these infections are ongoing. So what we're dealing with is a kind of constantly changing data set that we're, in a year's time will dramatically change. So what we want to do is use sufficient phylogenetic methods to inform us on the state of our data now, and then reapply these in a year's time, and so on. So rather than absolutely working, trying to get the true tree, you're just trying to get sufficient insight and trying to maximize your information from your current data without necessarily understanding every aspect of it. So these Leishmania donovani is one form of spectrum of parasites that infect people across the world. There are, I think, 30 types of Leishmania species, and they infect people in 88 countries across the world. And Leishmania donovani causes a version of the disease called visceral leishmaniasis, which is a particular public health interest because it disseminates to internal organs and is often fatal, but only after a prolonged period of maybe 10 years, during which it can be transmitted between hosts. This disease puts 190 million people at risk of infection every year and kills 50,000 people annually. So clearly there's a strong medical importance into understanding its molecular epidemiology. Although it's transmitted primarily by flies, in most human hosts it's asymptomatic. So it's clearly a very complex disease. Another feature of it is that different types or different species can hybridize, even though they might be very, very different at a nucleotide level. They can somehow produce viable offspring that give rise to new types that infect new people. And so we also want to get some sort of grip on how this establishes itself. And in that context, here are traditional definitions of what is a species and what is a population are not as clear, and it's something of a sliding scale in this particular example. So we were interested in Leishmania donovani in particular, and we compared initially this species to other Leishmania species who have their genome sequence. The genome sequences of these species have about 32 million characters. You can see there are some that are quite similar, like in Phantom, and there are some other ones that are quite divergent, which are useful for inferring the ancestral state of different alleles that we sequence, in comparison to our main focus, which here is donovani and also in Phantom. So we were looking at samples from regions with endemic levels of the disease, and wish to use both the interspecies information represented here and also the diversity within these different populations. So we initially aligned the genomes by starting at the most conserved segments, the genes, and iteratively expanding these regions to maximize the orthologous segments between the different genomes, so that we could essentially get whole genome alignments for these species. So in a conceptual form, you can see a sort of a representation of the interspecies diversity, where we're focusing on donovani and in Phantom in particular. So this tree represents how little diversity in comparison to the other species are this node, even though the genomes themselves are 32 megabases. So we've initially looked at samples shown on the bottom here in the region of the Terai region of Nepal and the Bihar region of India, because although they're geographically very close and genetically very similar, they have very, very different phenotypes and responses to drugs, and often give very different treatment outcomes for patients. We also tried to sample, or in the process of sampling, strains from across the world, shown on top from Europe, from Asia, from Africa, from the Middle East, to sort of give us a more firm context in which to place our phenotypically diverse sample set. So initially we created a reference genome for Leishmania donovani, and then we sequenced a whole 17 different strains from this Terai and Bihar regions and mapped this to the genome sequence. We performed very thorough variant detection processes in order to make sure that what we were inferring was a real variant and was present often in multiple samples in the population, so that we could look at the distribution of SNPs and not only look at our evolutionary history but also look at what portions of the genomes were under selection in this population. So when we look at the diversity within the sample, we can see that when we draw a median joining phylogenetic network, that there's evidence here, and here of multiple origins of resistance to different treatment drugs, even though this network here only represents about 400 protein level substitutions that are the only variants in these 17 genomes in a 32 million base pair genome, suggesting that, or demonstrating just how homogeneous these parasites are, and yet how their phenotypes are very different. So we try to expand our analysis to some degree by doing SNP genotyping, which rather than doing whole genome sequencing, looks at individual sites that are known to be variable across the genome, and we just looked at 130 of these in 38 strains as a sort of interim measure to get a grip on global diversity. So we sequenced not only our strains from India and Nepal, but also a few from around the world, from Africa, Asia, Europe, and Sri Lanka. And we found some clear structuring of the different samples from Nepal and Bihar, shown here in the different colours that were assigned as different populations using a Bayesian clustering algorithm called structure. One interesting observation was that some of our Nepalese strains, shown here, clustered with the divergent samples from Sudan, Ethiopia, Kenya, France and China, and also were quite separate from a strain we got from Sri Lanka. This was a novel result because previous analysis that had used microsatellites, which are kind of a very, very short 2 or 3 base pair tandem repeats, had shown quite a different picture, where there seemed to be no variation, shown here up top, and that our Sri Lankan strain clustered with the other samples, and this divergent lineage I pointed out on the previous diagram clustered with our Nepalese and Bihar sample set, illustrating how if you take different genetic markers from the genome, you'll get different phylogenetic pictures of the diversity. And clearly it's likely that there are very different histories operating with within different segments of the genome, and these obviously have implications for patients. In this particular instance, this Sri Lankan strain that separates these 2 strains from the other strains causes a slightly different form of the disease, cutaneous leishmaniasis, which is less severe, causes lesions on the skin. In contrast, this pair and the rest cause the visceral form, which is disseminates to internal organs. So clearly, this is an interesting kind of problem as to why they cluster differently using different segments in the genome, and also why they cause different symptoms in patients. This is just a representation again of the differences we can see between the two different approaches. Again, using structure. So this is again a network of these 38 strains that we use the SNP genotyping platform to look at, and just to give you an idea of how divergent these two samples that I pointed out before are shown here from the rest of the Nepalese and Bihar strains shown here. You can see that they're quite different, and they're from again this very, very close region, about 200 square miles, and yet they're clustered quite different clearly with the African and European and Asian samples. This is of clinical interest because these two samples were in fact sampled from a region without endemic visceral leishmaniasis, or they were taken back in 2003. But since the disease has spread into these regions, and they've become these type of parasites have been found more frequently. And obviously, if you've got a divergent parasite to which there's little host immunity coming into a region where there's frequent treatment using drugs, it may become the dominant type, and this is what we might be seeing in current time. So in terms of our plans, we hope to expand this analysis of these two Nepalese and Indian populations shown here, since these are slightly divergent, with strains from Africa, Europe, and so on, as a kind of to get a sort of an idea of the population-wide diversity from spanning Europe and Asia. But the evolutionary history of these different strains is likely to be complex because divergent lines can hybridize. So we expect hybridization events represented by these red arrows to have occurred, but also at some point in the evolutionary past, these different lineages would be geographically separated, so gene flow would stop. So we're interested in trying to get a grip on the evolutionary origins, and then consequently the migration of recent strains, as say people from regions with endemic VL from Nepal move to Africa, or Africa to there. In that context, when we look across the genome, we're expecting to see different signals of coalescence types, which might give us some sort of insights into which particular portions that cause virulence in host, or to what extent they come from populations in Africa or Asia. A second problem that we haven't necessarily solved is that we traditionally look at single nucleotide mutations only, but in these parasites we see dramatic genome-wide changes that are more complex. So we see structural variants, insertions, deletions, which might just be a few base pairs, but also might span anything from a single kilobase to 100 kilobase of DNA. We see copy number changes that they might have, receptor genes in particular might be replicated as some sort of tandem array that changes from generation to generation. We see entire duplications of whole chromosomes, which is quite different to what our traditional expectation might be of very static genomes. The genomes in these parasites vary wildly, and obviously this has large implications for how we might interpret this data. And fourth type of variant we find are episomes, which are extrachromosomal fragments, maybe I think from 20 to 50 kb, where a particular region of the genome has been duplicated, has recombined, and then is replicated at a very high rate by these parasites. So you may find that you have an extra chromosome fragment that might exist at 30 or 50 copies in an individual parasite. So obviously trying to combine these different variants we find is a challenge to which we don't have a solution yet. Just to get an idea of the extent of copy number variation, you can see here we have the different chromosomes in Leishmania donovani, some different strains we've sequenced, and shown at the top left you can see a color code showing the number of chromosome copies we have, which are roughly approximate integers. And you can see that they vary a great deal. Using structural variation data, we can also, to some degree, construct phylogenies that are very similar to the story we get from SNPs, but only to the extent that linkage between the structural variants and the SNPs is not disturbed by historical recombination events. And although there are proposals of how to construct phylogenies using structural variants, it is a challenge, because rather than being able to equate one insertion to being one deletion, if one of each is 10 base pairs and one is 100 base pairs and another is 1,000 base pairs, how do you equate them? So this talk was geared towards just giving you an idea of how messy and complex real-world clinical data can be, and how phylogenetic tools can be used to some extent for identifying divergent lineages, but also what challenges are ongoing and can be directly applied to clinical data. In particular, can we use the differential clustering of different strains towards finding regions that are of particular relevance to disease pathologies and drug resistance? Can we use this to look at hybridization events? How do we coordinate the different variants we detect in the genome? And also, how do we can we use phylogenetic methods to inferring ancestral recombination events that might have occurred within populations, and also as hybridization events between ancestral types. So finally, I'd like to thank different people in the Wellcome Trust Sanger Institute, but also our partners in Antwerp and in institutes in Nepal and India as well, who have put together a very thorough analysis of these parasites.
Thank you, Tim. Are there questions?
I've got a comment and a question. The first one is, I'm working with colleagues at Rothamsted Research, and they some of them work on a particular type of fungus, which has very different sort of patterns in different kinds of hosts, and it all boils down to little micro chromosomes, which seem to sort of, they seem to sort of be transmitted somewhat independently, and the way that they infect their host depends on these very small micro chromosomes. It depends which ones they've got. Now, it might just be the same sort of story here, that these extra little bits of chromosome seem to be highly replicated in some of these species. It might actually confer their sort of the kinds of cells that they attach to within the human. So just a comment. But the question I had was, you've looked at these at least 17 whole genome sequences, 17 species, and so presumably you can look at that and get some idea of the hybridization patterns, if you can see them if they've occurred between these different species. I mean, are they like little bits which kind of swap in and out of some of the chromosomes, or are they like two, one end of the chromosome comes from one species and the other one comes from another species? Can we actually see the patterns of hybridization and what sort of scale do they operate on?
Well, within our 17 genomes, we find that there are very long linkage blocks where you get 50 kilobases that are shared between one set of strains. You've extended haplotypes that you get a single set of mutations in one set and a single set in the other set, and they don't appear to be very different. But we're in the process of looking at European strains and comparing these to our 17 strains from India and Nepal. So we have some expectation that we will see these blocks again, but we're not sure exactly to what extent they exist yet, just because we haven't performed the alignments. So it's kind of an ongoing progress, but that's a key interest to identify the recombination breakpoints.
Okay, I think due to time, we'll move on now to